Hello, I am talking about and I'm presenting my findings on the enzyme lab. An enzyme is a protein that catalyzes a chemical reaction by lowering the activation energy needed to carry out the reaction. This is very important because they speed up reactions by lowering the reactant's activation energy. The enzyme we focused on is trypsin. Trypsin is a proteinase that is typically found in the small intestine of humans, and it's secreted by some forms of bacteria. It catalyzes the hydrolysis and the breaking down of peptide bonds. It can be found in humans around the pH level of 8. This is because proteins denature at certain degrees and salinities. Using the information from the previous experiment, we were able to predict that the optimal temperature for enzymatic activity would be about 50 degrees. The substrate being used was azocasein. It is a chemically modified protein by adding sulfonylamide groups to the milk protein casein. They absorb light at 440 nanometers. We use temperature as our independent variable. The variables we used were between 0 and 90 degrees Celsius. In all cases, we had 500 microliters of azocasein and 50 microliters of trypsin. Every reaction was incubated for 20 minutes at the different intervals, 0 degrees through 90 degrees. After incubation, the reaction is stopped with TCA, or trichloroacetic acid, and this precipitated specific macromolecules, including the enzyme and undigested azocasein from the reaction. Those participated molecules were removed through being centrifuged. We then put it in the centrometer, and its absorbance is measured at 440 nanometers because that is the absorbance of the molecules of azocasein. Since our digested azocasein samples produced a darker red and orange, the undigested samples, the higher enzyme activity, produced higher absorbance values. We used a control variable. For us, it was a room temperature sample with the same salinity as other samples. We just didn't use trypsin. Our negative control had 50 microliters of water. We replaced the trypsin to see if the substrate would be digested without the enzyme present. This would mean our experiment would be false. When going through the experiment, we used a pH buffer of 7 with a salinity of with a certain salinity. Using a micropipette, we placed 450 milliliters of enzyme and specific buffer into cuvette. We shake the cuvette until the contents are thoroughly mixed. We took the cuvette with the sample and placed it into the temperature-controlled chamber. We let it sit for 20 minutes, and then we took it out of the chamber, placed it in the centrifuge, highest speed, for 2 minutes. After that, we took it out, we pipetted the remains out, and put it into the spectrometer. We then recorded the readings in nanometers. And then we repeated these steps over and over again until we had the amount of samples that we needed. Our results are shown here. The digested in the absence of trypsin was found to be significantly lower than that with the enzyme present. Our results also showed that the rate of enzymatic activity slowly raised until the optimal temperature at 50 degrees. And after the temperature continued to increase, it slowly decreased. To determine the level of significance, we did a bunch of t-tests in comparison to the absorbance value. To test the null hypothesis, the temperature peak was significantly different from the other temperatures. We also had absorption levels steadily increase from temperatures 10 to 40 before peaking at 0.58 micrometers at 50 degrees. The average standard deviation for tests was very low, ranging from this number and at the lowest to this number at the highest, statistical analysis was done through t-test. Absorption at 50 degrees Celsius was compared to every other temperature. Absorption at 50 degrees was found to be significantly different from absorption at every other temperature except for 60 degrees. Our variability between our trials measured by standard deviation. The greatest difference between enzymatic reaction was between 40 and 50 degrees Celsius. The next biggest happened between 60 degrees and 70 degrees when it started to decline in enzymatic activity. Our enzyme activity was calculated by using absorbance readings of the digested azocasein. The statistical analysis was done using the t-test. The activity levels were tested at temperature level. To define significant difference, we wanted to find a p-value less than 0.5. We hypothesized that the enzymatic activity would increase exponentially until it reaches its optimal point at 50 degrees Celsius, where it would then begin to decline in activity. To help ensure we had a large number of samples to choose from, we used 8 samples, each tested 5 times. In total, we had 40 samples. 1 negative and 1 positive control tested 5 times. This showed us that if there is an outlier, that all the other samples would balance it out. Our t-test shows significant differences between all values except 1, which proved that we could not support our hypothesis because one of them did not work with it. Our group, as well as groups F and E, used temperatures as their independent variables, and table A and C used pH as their independent variable. Group C and A, the ones that tested pH, found that optimal pH for enzymatic activity is a pH of 9. From another class, they found that the highest level of enzymatic activity when it comes to salinity is at 0 molar. So the enzyme would react the best when it's in a pH buffer of 0, a pH buffer of 9, a salinity of 0 molar, and at 60 degrees Celsius. There are a few ways that variability could have occurred. Pipetting errors, mistiming incubation, mislabeling, or touching the pipette to a surface before cleaning. Between our first and second experiment, there were big improvements. We had many replicates that allowed for us to see consistency between all samples. For the future, my lab group could have done more temperatures, and instead of doing every 10 degrees, we could have done every 5 degrees to find an even more specific temperature for the peak and the decline. Our results showed that trypsin had a clear impact on the digestion of azocasein, and the effectiveness of its digestion had a clear correlation to the temperatures. For the future, we should ensure that every class has at least one table that is one of the 3 possible independent variables. This will allow for us to communicate with them and to compare our results to find the absolute optimum conditions for the enzymatic activity. This bacteria will grow better in these ideal conditions. Based on that bacteria tested, the optimum values would be a pH of 9, a salinity of 0 molar, and a temperature of 60 degrees. Our discussion at the end was, it was hypothesized that absorption would increase until reaching the optimum temperature between 10 to 70 degrees Celsius. Absorption peaked at 50 degrees at 0.588 nanometers. These results were consistent with the hypothesis. There is no significant difference in absorption at temperatures 50 and 60 degrees. Absorption at all other levels was significantly different. These findings suggest that the optimum temperature for enzymatic activity is somewhere from 50 to 60 degrees Celsius. I hope you all enjoyed my presentation. Have a good day.